What do you think of when I talk about growth factors and hormones? For many people, the first thing they think about are the factors that regulate how tall you get and hormones that become active when you hit puberty. Here, I want to show you that hormones and growth factors play much greater roles than that and that they affect many processes at all stages of your life. I'm Janine Post of the Developmental Bioengineering Group at the University of Twente, and growth factors and hormones are at the core of my research interest. Organisms have specific needs in responding to their environment. For example, we can think, I'm hungry, I will get some food. Within the body, our cells have similar needs and need to communicate with the rest of our bodies about the needs for nutrients. Similarly, cells in the body and the body as a whole needs to be able to respond to danger. This response is also mediated by hormones, in this case epinephrine or adrenaline, that tells our muscles to run and our liver to free up some glucose as a nutrient source for our muscle. When we look at growth, then we know we started as single cells, but by now we have trillions of cells. Not only do we have many cells, we have many different specialized cells with different functions. So we have grown, and this growth had to be properly managed. For example, the fact that my both legs are the same length is controlled by hormones. So growth factors and hormones have to coordinate their efforts in tissue formation, tissue maintenance, and response to environmental changes, very much like rowers coordinate their efforts for balance and maximum speed under different weather conditions. So what makes cell communication so important? Well, cells receive many simultaneous signals that tell it what to do. And we know that disruption in this communication can lead to diseases, and that disruption of communication can happen at the level of DNA, RNA, and protein. If we identify the changes leading to the diseases, we have potential targets for intervention. And intervention, in turn, can be a possible therapy. Cell communication occurs through a process that we call signal transduction. In this, a chemical signal is recognized by specific receptors that are mostly located at the cell surface. These receptors relay the signals to intracellular proteins, which results in different cellular effects. These include processes such as cell growth, proliferation, cell migration, and cell differentiation. Cells use extracellular signal molecules to communicate with another in various ways. Hormones that are produced in endocrine glands are secreted in the bloodstream and are distributed widely throughout the body. Examples are adrenaline and cortisol. Paracrine signals are released by cells into the extracellular environment and act locally. These include all cytokines and growth factors, for example, epidermal growth factor. Neuronal signals are transmitted electrically along a nerve cell axon. When this electrical signal reaches the nerve terminal, it causes the release of neurotransmitters, which are bound by receptors to adjacent target cells. Examples are acetylcholine. In contact-dependent signaling, a self-surface-bound signal molecule binds to a receptor protein on an adjacent cell. Signaling molecules are greatly diverse in terms of chemical structure. Some are small, hydrophobic molecules. Others are small or larger polypeptides. The same signal molecule can induce different responses in different target cells. Take for example acetylcholine. Acetylcholine binds to similar receptor proteins on heart pacemaker cells and salivary gland cells, but with different responses in each cell type. The heart pacemaker cell fires more slowly when acetylcholine is bound, while the salivary glands secrete saliva. Skeletal muscle cells produce a different type of receptor protein for the same signal molecule and contract when acetylcholine is bound. Acetylcholine has a simple chemical structure, but is very versatile. It is known to function in the enhancement of alertness and is necessary for being able to stay focused and for learning and memory. Cells receive many signals that integrate via a so-called signal transduction network that integrates and modulates all these signals. To describe processes such as cell differentiation or disease in time, we make use of dynamic computational models of signaling networks. In this example, we made a computational model of cartilage, in which we investigate the signals regulating osteoarthritis. In the model, the signals activating the transcription factor SOX9 are slowly turned off, while the signals regulating the transcription factor RUNX2 become active. In that case, osteoarthritis occurs. The advantage of using computational models is that we describe the whole process and predict what therapeutic options are available for repairing the tissue. Research on cell communication is performed at all levels of signal transduction, from the signal to the receptors, from intracellular signaling molecules to effector proteins. Because mutations occur at all of these levels, and the effect of a mutation cannot be predicted, a mutation can lead to less activity, but also to more activity. Some newer drug treatments function by blocking the growth factor binding to its receptor. If you want to investigate whether this treatment is effective, you can quantify growth factor binding to a cancer cell in presence and absence of that treatment. In this case, we made a fusion protein between the epidermal growth factor receptor in green and expressed it in live cancer cells. To these cells, we added epidermal growth factor coupled to a red fluorescent quantum dot. After addition of the signal to cells, we see immediately extensive membrane movement and changes in the shape of the cell. This method enables us to quantify the activity of the receptor in presence or absence of certain cancer treatments. After activation of the cell receptor, intracellular signaling molecules become active. This leads to activation of transcription factors that translocate to the nucleus. This can be observed by live cell microscopy. After translocation to the cell nucleus, the transcription factor regulates gene expression of a specific set of genes under its control, thereby regulating the cell's response to the signaling molecule. At the level of cell responses, one can look at cell division. In this example, we observe coordinated cell division in early fruit fly embryos, in which a DNA-binding protein, histone 2A, is coupled to green fluorescent protein. This synchronous cell division is tightly regulated by growth factors and hormones. Has this lecture made you curious about cell communication? Check out my YouTube channel for more mini lectures or sign up for one of my courses. See you soon!